Now it's my pleasure to introduce Dieter Beule, well known to many of you here in Berlin and also beyond, who holds a professorship for Translational Bioinformatics as part of our section Translational Sciences and Applications. Now, in very simple terms, Dieter Beule aims at making complex molecular data usable and understandable for clinical practice, irrespective whether data arise from technologies and techniques like genome sequencing, single-cell analysis, or proteomics quantification. He has already been heading the core unit, Bioinformatics QB, a joint research initiative of the BIH, the MDC, and also the Charité. His background, again, is highly interesting, and again, reflecting a very agile career development. Dieter Beule studied physics at the universities of Freiburg and Sussex, as well as at the Technical University here in Berlin. In 2004, he received his PhD from the Humboldt University in statistical physics. In 1998 already, he started work as a bioinformatician at the Humboldt Institute for Theoretical Biology, where he conducted research on expression analysis and transcription factor binding sites. From 2001 to 2014, he successfully founded, so for more than a decade, and established 2 companies in the fields of bioinformatics, offering services, softwares, and in vitro diagnostic devices. In 2015, Dieter then established the new core unit Bioinformatics, and since then he has also led the high performance computing unit. So once more, we have a beautiful paradigm of agile career development, A to B and back to A, in Dieter's case involving the formation of new companies. Today, Dieter is involved in numerous large-scale research networks, such as the German Human Genome Phenome Archive, GA, within the National Research Data Infrastructure, NFDI, and the German Network of University Medicine, NUM, and also the German Genomics Initiative, Genome.de. So Dieter, we look forward to your presentation.
Yeah, thank you, Christopher, for the very kind introduction. So I wanted to start out here with taking a look at the omics data revolution that we all observed in recent years. So usually this is discussed in terms of data amounts. I would rather say it's about diversity and complexity of what you can do. I mean, you can use all the creativity in research and somehow transform it into some sequencing experiment, and that poses the challenges in the analysis. So we measure millions of times, we see all the funny noises and long tails and rare errors. We have to kind of make stable and reproducible workflows to analyze that. We have to capture all that creativity in metadata. And then even if bioinformatics can reduce that data amount from terabyte or gigabyte to megabyte or kilobyte, it still needs also the biomedical expertise to further interpret and analyze that. So interpretation, integration, interdisciplinarity are really key. So translation, what does it mean in bioinformatics? Not that I should have translated this slide, so sorry, I just noticed. Translation really means that we drive this methods, this bioinformatics methods, for me, down this chain from like very scientific, dynamic, experimental, in getting the technologies stabilized and optimized, and getting then systems to empower people to do analysis and develop processes until we really come to standardized, automized, established procedures. So usually I also emphasize very much that getting your data sorted and getting them organized is very important, but I skipped that today. So for today, I would rather try to look at a few tasks that I think are important in this realm and discuss them in the context of some tools that came out of my research. So I start with something from mass spectroscopy, which actually now takes just the same way as sequencing. So many different methods are evolving, many different things are coming from this direction. It's moving into the clinic. So Mstars project, for example, which I'm also involved, is a prime example of that. So, and here, this is something that emerged from a collaboration with Jennifer Kirwan from the metabolomics unit. So we look into liquid chromatography mass spectroscopy. So basically, you have your samples, you get a two-dimensional measurements in retention time and MZ ratio, and you get thousands or 10,000s of peaks for the different chemical compounds. And if you want to do that over clinical size cohort studies, then there is a big data analysis task there. So of course there are tools, there are great tools there which kind of capture like the experience of people who developed them for decades basically, but all of these have a tendency to overpick things. So and this impedes very much downstream analysis, and used to be like a very interactive human kind of manual peak correlation thing in the end going on, which also impeded what's laborious and also impeded reproducibility. So we try to improve on that, so to make this more scalable and more reproducible. So one idea, of course, would be to replace everything there with machine learning. There are tools out there that try that, but because the old methods are really good, they don't really succeed so much. So if we came up with integrating something machine learning based into existing workflows and really just focusing on the specific task, can we use the expert knowledge on what a good peak is and what a bad peak is to filter it before we further downstream analyze? And so that is, it's a machine learning exercise. We have to think about how to create an architecture for the feature extraction, to create the classifier. If you want it to perform, you also need to think about proper abstraction of your data. And I don't want to go into the details here, but this is like a nice project that we developed interactively with Jennifer. And then once we have this, we can actually train the system. So we can use experts and have them bring in their expert knowledge and move it into the neural network, the deep neural network that we are using there. And an interesting thing is that, first of all, it learns this very well. This is the ROC curve showing on the right-hand side. And the second thing, it's actually quite transferable. So if you trained it on one study, it will work on another study, even if you do slightly different protocols or slightly different machines, and that is very powerful. But if then it isn't good enough, we also implement a transfer learning thing. So you can improve on your models, and this way it becomes very, very agile, usable. So this slide is showing comparison with best practices, so with the conventional tool, with full machine learning based quantification tools. And what you see is here we analyze a data set which is a fully complex matrix plasma, but we also spiked in like 80 something components with different concentration and see how much we can recover. And in fact, we very much outperform the machine learning tools and the existing machine learning tools, and really get a lot, factor 7 here, in some cases less peaks that are actually needed for processing compared to the conventional tools. So that's a big step forward. Maybe even more interesting is how this performs really if you go to the detection limit. So here we start out with a spike in sodium. This time it's not a complex mixture. It's really only 41 chemicals in there, which are near the detection limit, and we further dilute them by up to a factor of 100 and see how many we can still recover. And the most interesting plot is down here, E, which is really about the migration if we go along the dilution time. It's like a voter migration plot in the election times, and you can see that there is a consistent migration from like the green ones that are well detected to the ones that where the quality then becomes questionable, until finally they disappear into the undetectable rearm. So that is very very useful, and by now that tool is, so here's the preprint, but actually as of yesterday, it was accepted in analytical chemistry. So we have here a tool which really plugs in nicely into existing workflows. It's really performant, so analysis is minutes, and even if new training is just a few hours, we made it in such a way that it can be widely used and easily installed, and we think it will be comparable with and compatible with many use cases, and we're now also exploring that in Mstars. So that would be like an example, new methods for new data. And I mean, the whole translation process is not a big leap forward. It's more a lot of small steps. And I have a second small example, which is dear to my heart, which is really new methods for old data. That is something, uses something that we learned for copy number change analysis in genome, in big genome cohorts, and we brought it back to Panelistic analysis. So we are in the realm of rare diseases now. So there's a lot. For the longest time, this was analyzed with Panelistic data, so where you just focus on specific genes, we have some suspicion which there would be, and they work nicely for like small variants, but there was never good methods for calling copy numbers based on that. And so, but with the emerging of better methods for cohort analysis, and with these big historical data sets or legacy data sets being available in the clinic, we started out an endeavor and look into that. And the challenges here really, it's very noisy, different kits. It's not well documented many times which kit and which batch precisely was used, and so but that can be overcome with two component things. So some unsupervised machine learning that really classifies the things into cohorts that can be analyzed together, and these new methods that emerged from genome analysis, which really tries to look across cohorts, the sequencing depth across cohorts, and tries to make a model about that. And here on the lower right side, you see this red plot, and it's targets in genome, ordered in genome position and samples. And so you can see, for example, on the right-hand side, there's a pure sample. On the middle, there is something with some quality issue, the region D there. There is also maybe a target highlighted as B there, which is poor. But if you see something like A, that is the sequencing depth is consecutively changed over several targets in your panel. That is a copy number change, and that is the kind of things that you want to see. So we looked into this, and was initially a bachelor thesis, but we then invested some extra time to make it nice. We have very competitive sensitivity here, and with this machine learning part, we also really very much increase the specificity, and so we have by now a lot of qPCR confirmation of the findings that we had. We have actually solved all cases, and so it actually gave people a diagnostic, even if it's legacy data that's set in the files for some years. And that I think is a nice story. It's now under review in bioinformatics, and we next look into, can we also do sensible things on cancer panels here. So I'm probably talk too slowly or have too many slides. So next thing, I want to go into like interdisciplinary exome and genome variant analysis. So this is, as I said, there is, even when the bioinformaticians are done, there's a lot of things to do for the clinical people. And so we worked a lot on this for many years, and there is this publication in Nucleic Acid Research, WAFISH, our software that empowers researchers and biomedical people and clinical geneticists to analyze cases and cohorts. And I don't want to walk you through all the features here, but rather I want to highlight the analysis approach that is really empowering user-user collaboration. So within the teams, people can like put cases aside, they can publish things, results on ClinVar, they can reanalyze cases if new databases are coming up, and the other really important interdisciplinary task there is the interaction between a clinical geneticist and their way of analyzing cases, and us developing new features and implementing new features. And that really made things strong, I think. So actually, even this slide is not by me, but it's by Nadia Imke, one of our collaboration partners, important collaboration partners in medical genetics. So we have a really nice solution there. Now we have the next challenge, actually, sustainability. So we have Warfish is starting to get popular among other clinics. So we have 5 installations across Germany, 1 in the US, a few in Eastern Europe. So my next and final slides are really about merging open source and translation. So we are bioinformaticians. We believe in open source and Glassbox, and the progress in genomics is because of open source, I think, very much. So open source software is free, as in free speech, so no locking with some vendor that then can enforce things on you. Free as in free beer, everybody can ask the same, everybody can change, but also free as in free public. So you have to take care of it and have to take responsibility for your installation. And so if we want to make that sustainable, we should think about the free puppets, not only the free beer thing. And that the needs there are really that, okay, if third party wants to use that, they want some service level. They want someone to maintain the thing. If there is an update, if there is a bug, if Windows changes, whatever. And then also the emerging user needs should be embraced. Yeah. So there's many good practices out there. All of you are using it. All the internet runs in open source software like Linux and MySQL and things. And also in your work, like RedCap is very popular with studies, and GATK is one of the core things of all the genome sequencing. And on the right-hand side, there are 2 things where actually I spend a lot of money to keep the consortium running, because those softwares, Slurm and iRod, solve a lot of my problems that I'm having. So when also, I see this very much as there's a positive commitment of society. So we want digital sovereignty, and the coalition contract even said public money, public code. So I think this is the right way to go. So the status quo really is we move forward on this effort. So we have put our project out there, and we have all the test automation to ensure the quality of the issue tracker. We have really good installation support by now, and we started a special interest group for developers, also for third-party developers and for third-party users, monthly and weekly, and formalizing the development process there. And we have, like with University Bonn, they have an interest also in implementing new features, and University Clinic Aachen, they are more like an only user to have this kind of proof of concept processes being established. And more formalized, we're also moving really into proper life cycle documentation, so that eventually this can be used in clinical diagnostics. And we'll probably need to talk to Mike Friese how to create a legal entity that then can actually also accept third-party money for providing certain service level agreements and things like that. But when we get this done, this will be a really great blueprint for moving digital innovation fast into clinical diagnostics. And with that, I summarize my things. So I hope I gave you some impression what the things are going on in this translation along the line as I sketched it. And I want to thank my team and all the collaborators that I mentioned along the way, and look forward to your questions. Thank you.
Thanks a lot, Dieter. It was perfectly clear that there is a big challenge behind the sustainability of the open source world. And however, it's absolutely clear that we really need this to be developed through public funding and by public institutions. I don't see a question right now from the audience. So my question would be, through what level of organization do we have to go to divide all the tasks that we have in this field? I mean, there are certain institutes who are driven by individuals like yours and your team, who can overtake some of these tasks, but who has oversight of what is needed for the entire community, and how will tasks be distributed in the community?
Yeah, so I presented this idea also a bit more elaborate in the Arbeitsgemeinschaft Genomdiagnostik last week, and people are very interested, and we have also been invited to Austria and also identified some potential other users. And my current idea is really that we start a something like a GmbH or something where then people can fund for certain services, service level agreements, and that in turn can fund some staff that can continue working on the system. So that is the general idea, and that is kind of what also other organizations are doing. I mean, like the ones I mentioned with Slurm and IROTS and RedCap and the like. So that seems to work for academic software in general. Here, what we have is the challenge of the certifications or of the quality management. So,
but in the end, it could be something like a European platform or something international driven by public resources.
Yeah, I mean, midterm, yes. I mean, first of all, we probably have to show that it works in diagnostics. So that would be my first milestone.
Yes. Good. Okay. I don't see another question coming up, but I'm sure people know how well they can use your support and how important your support is. That is absolutely clear. So thanks for presenting